All right, now as we go into the details of cell division and how the genetic material is copied and divided, it's important to introduce maybe some new terms when we're looking at these condensed chromosomes in the cells that are going through active division. So let me draw this out before we talk about the information on this slide. Okay, so a eukaryotic cell has linear chromosomes. And when it's condensed, right, it might look something like this. So this is just one unduplicated chromosome. That's one chromosome. Now the cell wants to go through cell division. So it says, okay, well, I need to make an exact copy of all my chromosomes, and I need to give one copy to one daughter cell and the other copy to the other daughter cell so that both daughter cells have the necessary information that they need to function. And so what the cell will do is it'll go through DNA replication. Draw an arrow, DNA replication. And what it does is it takes this single linear chromosome and it makes a Xerox copy. And the extra Xerox copy actually sticks to the original copy, essentially. We'll talk about DNA replication later. It's a little bit more complicated that both copies have some old and some new DNA. But these two copies now of the DNA are called sister chromatids. So this guy right here and this guy right here, they're both sister, all right, two sister chromatids. All right, now this whole structure though, combining the two sister chromatids, is still one chromosome. We will say this is one duplicated chromosome. All right, so before DNA replication, we have one unduplicated chromosome, and after we make a copy of the chromosome, we still have one chromosome. It just has two copies stuck together. The two copies are called sister chromatids, and the full thing is one duplicated chromosome. You'll also notice that here in the center, and that center can move, it doesn't have to necessarily be in the dead center for a chromosome, it can be towards either end too, but I've drawn kind of a narrowing of the chromosome. This narrow part is called the centromere. I like to call it like the narrow waist of the chromosome. This is where the DNA is really tightly condensed. This is also where the two sister chromatids are held really tightly together. So a new term here, centromere, is that narrowing of the chromosome somewhere along its length. Okay, so if we go back now to this particular slide, we can see now not a cartoon diagram like I drew, but an actual electron micrograph of a duplicated and condensed chromosome from a cell that's going through cell division. So here's one sister chromatid on the left and another sister chromatid on the right. You can see the narrow waist here. This is the centromere. And the sister chromatids are held tightly together. They're held tightly together by proteins that are called cohesins. So if we go back to our little figure here, cohesins are probably going to be like, I like to think of them as little rubber bands that are holding the two sister chromatids really tightly together. All right. Okay, so the next thing we're going to do is we're going to go through just a very generic example of what mitosis is going to look like in a cell before we dive into all the details. So here we have the most simple of cells. It's a single cell, and you can see it has a single chromosome there in the center. The centromere here, the narrow waist of the cell is there. On either side of the centromere, we have what are called chromosome arms. One might be a little bit shorter, one might be a little bit longer, but regardless, we have a single DNA molecule here making up that chromosome. So this cell now says, I want to divide. The first thing it needs to do is it needs to go through DNA replication. And so the cell will make an identical copy of its chromosome Now here in this cell, we still have only one chromosome. It's duplicated, made out of 2 DNA molecules, and we call those 2 copies sister chromatids. And then through the process of mitosis, that cell will separate the sister chromatids, give one copy to one daughter cell and the other copy to the other daughter cell, and each daughter cell now is identical to the other and also identical to the original parent cell genetically.